The overall goal of this method is to generate parabiotic zebrafish embryos, which can be used for studying cell intrinsic versus cell extrinsic functions for candidate genes of interest.
This method can help answer key questions of cell autonomy. For example, in the hematopoietic system, whether defects in HSC migration are intrinsic to the HSCs or the niche microenvironment.
The main advantage of this protocol is an improved ability to successfully generate parabiotic zebrafish embryos.
Generally, individuals new to this method will struggle because the developing embryos are extremely fragile and easily damaged.
Visual demonstration of this method is critical, as the surgical stitching steps can be difficult to learn because they require a high degree of accuracy and precision.
Here we provide step-by-step instructions to generate parabiotic zebrafish embryos by surgical fusion of developing blastulae.
This method demonstrates how to increase efficiency of parabiotic fusions and enhance survival of conjoined embryos.
After preparing reagents and tools according to the text protocol, prepare 2 to 3 modified Pasteur pipettes by briefly heating the end of the pipette over a Bunsen burner. Then, using large forceps, while it is still hot, bend the end of the pipette approximately 45 degrees. To ensure that there are no sharp edges that might damage the embryos, heat the end of the pipette over a Bunsen burner for 1 to 2 seconds. Using plasmid DNA, mRNA, or morpholino, microinject embryos within 1 hour of collection and allow them to develop.